Thank you, and welcome to everyone joining our call today about the WAVE. We wanted to say, first of all, thank you all for joining. We know that this is the holidays, and so this morning is early for some of you, late for many of you, but we're really thrilled to be here with Jeff Smith and Mike Penzer. This is being brought to you as a collaboration with the World Duchenne Organization and PPMD. Alex Johnson and myself are board members of World Duchenne Organization, and so we're thrilled to join you. We've all heard the devastating news that WAVE is terminated the program. So first, what we're going to do today is hear from Jeff and Mike, the CMO of WAVE, to really have an opening statement. So I'm going to turn it over to you, Jeff and Mike. Please go ahead.
Sure. Thanks very much, Pat, and thanks to you and the team for coordinating the opportunity for us today. And thanks to everyone for being on today. So it's obviously been a challenging week, and certainly not the week that we hoped or planned to be having. On Monday, as you alluded to, Pat, we announced the difficult decision to discontinue the Suvodirsen program. This was frankly heartbreaking for us at WAVE and deeply disappointing that we weren't able to achieve the goals that we set out to achieve and make the impact that we hope to make with Suvodirsen. Primarily, though, our thoughts are with you in the community, particularly the families, the individuals, and the families that participated in our clinical trials for Suvodirsen. You made in many cases, transformed your lives to participate in our clinical trials, and we sincerely appreciate your contributions to our work. So for that reason and others, we obviously thought it was important to be here today. So thanks again, Pat, for having us. Mike Penzer, Chief Medical Officer, I'll turn it over to him to share a little bit more about the decision and what led to the decision. Mike?
Thank you, Jeff.
Yeah, thank you, Jeff, and thank you, Pat and Alex, for sponsoring this call. And good afternoon, good morning, and thank you to all the participants today for joining us. I speak on behalf of the entire WAVE team when I say that this outcome is completely unexpected and truly deeply disappointing. Before I get into the results, I just want to say we are sincerely grateful to be part of this community. And WAVE, our entire organization, has been shaped by your strength and resilience. While we made the difficult decision to discontinue the Suvodirsen program, we intend to do everything we can to fully understand these results and share them with the entire community to ensure that our learnings become your learnings and benefit further research. So as we announced this week, the open-label extension trial interim results showed no change in dystrophin from baseline, as measured by Western blot, with either the 3.5 milligram per kilogram dose or the 5 milligram per kilogram dose of Suvodirsen. Based on these results, we discontinued development of Suvodirsen, including both the open-label extension trial and the phase 2/3 Distance 51 trial. The interim analysis was from our open-label extension study. This was a global multi-center study, or an OLE, of Suvodirsen dosed weekly. 36 patients enrolled in the study. These were patients from our phase 1 single ascending dose study and received either a dose of 3.5 milligram per kilogram or 5 milligram per kilogram. A biopsy of each patient's deltoid muscle was performed prior to the initial dose in the OLE, and as of the date of the cutoff, follow-up deltoid muscle biopsies were available for 27 of the 36 patients. The follow-up biopsies were collected at 22 weeks for patients on both doses. In addition, there was an exploratory analysis in the OLE which included biopsies collected at 12 weeks after dosing from a subset of patients in the 5 milligram per kilogram dose. And this subset was designed to help us plan for the interim analyses that had been planned in the phase 2/3 Distance 51 study. And as you remember, that was all being done to hopefully adapt that study and minimize placebo recruitment. Specifically, among patients on the 5 milligram per kilogram weekly dose in the OLE, 10 patients had follow-up muscle biopsies at 12 weeks, and 9 patients had follow-up biopsies at 22 weeks, again in the 5 milligram per kilogram weekly dose group. Among patients in the 3.5 milligram per kilogram weekly dose in the OLE, 8 patients had a follow-up muscle biopsy at 22 weeks. So biopsies from both of these time points in both doses were part of the interim analysis of the OLE for dystrophin expression. Unfortunately, there was no change in dystrophin expression from baseline at either dose, 5 milligram per kilogram or 3.5 milligram per kilogram of Suvodirsen, or either time point. There was also no change in CK levels. Based on this observation, the lack of dystrophin and these other signs of effect, we made the decision to discontinue Suvodirsen and immediately stop dosing patients in the OLE, as well as the Distance 51 trial. All patients will have a follow-up visit, and no further muscle biopsies will be collected. There were no safety concerns or emerging safety signals observed at the interim analysis, so this decision was based completely on a lack of efficacy. On behalf of everyone at WAVE, I'd like to again extend my sincere thanks to the advocacy community, individuals and their families for their contribution to this program, as well as our colleagues at the Duchenne centers throughout the world whose guidance and dedication made the outstanding execution of these studies with the quality data that enabled the decision possible. We have a responsibility to all of you, most importantly to your boys and their families, who had courageously participated in this research to fully analyze and understand the available results from the OLE, and we do intend to share them at a medical meeting sometime the first half of this year. After the final data from the study have been analyzed, we are committed to enabling investigators to share individual patient results with each patient and his family. In addition, unfortunately, we're suspending further development of our N531 program, which was a treatment for patients with mutations amenable to exon 53 skipping. This was a particularly difficult decision. However, we believe it's the best interest of patients and families to prioritize the understanding of the Suvodirsen OLE interim results before we do anything else. So with that, I'll turn the call back over to Jeff.
Sure. Thank you, Mike. So as Mike just mentioned, we're going to be working hard over the next several weeks and months to analyze, understand, and then communicate back the results, so that, of course, we can inform further research and focus on incorporating those learnings into future research. I just want to, personally thank all of you out in the community for your support and guidance. Particularly over this past week, we have received just so many notes of thanks and notes of support, which we truly appreciate. This is a unique community, and we've, as Mike mentioned, we're grateful to be a part of it. So, with that, why don't I, Pat, turn it back to you, and we can address some of the questions that have come in.
Sure. Thanks, Jeff and Mike. I so appreciate that opening. It's, as you know, very difficult news, and we were also hopeful. We've believed in the antisense oligonucleotides, exon skipping technologies for a very, very long time, and seeing them come to the forefront has been exciting and promising and very hopeful to so many of us. So can we just start with the basic science and talk about the preclinical data? The preclinical data, certainly in the mouse model, looked really, really amazing as you shared the results with us over time. Were there other animal models you used, or can you give us a little insight into why you think the animal models were, so clearly positive with Suvodirsen, and why that didn't translate into the human?
Yeah, I can take that one. Well, unfortunately, we really don't know the answer to this question right now. I mean, as you noted, we had very strong data in human cells. We had great data in the MDX mice, which is the typical DMD model, and we even were able to amplify transcript in monkeys, which is quite a difficult task. The next several months, this will be a key focus of the work we're going to be doing. There's a lot of things that could have happened. At the basic level, it could simply be a problem of the drug getting from the blood into the muscle fiber. We know it was in the blood. We detected that in our phase 1 study, and even in the OLE study, we know it was in the blood, but maybe it couldn't get into the muscle fiber for some reason. It could have gotten into the muscle fiber, but didn't go into the nucleus, which is the place where skipping occurs. It could have been transcribed or otherwise changed from DNA to an RNA, but never made it to a protein, which is called translation. So a lot of things need to be explored. We're committed to understanding what happened, and once we figure that out, sharing that with the community.
That would be wonderful. And in terms of the assay to detect dystrophin, there are a number of different ways and presentations of dystrophin. Do you feel that using Western blot is the best, most appropriate to understand and detect dystrophin?
Yeah, we're quite confident in the assay. It was very sensitive. We'll share all those details when we share the data, but I can definitely say the assay was reviewed by FDA, was sensitive enough to detect extremely low levels of dystrophin. To know we didn't just stop at dystrophin. We looked at other measures of efficacy, including immunofluorescence, looked at changes in CK. We truly saw nothing. So we're quite confident in the decision.
Okay, thank you. And you've talked about the phase 1 study, and may I remind the audience that should you have questions, please stick them in the chat box. We have received questions prior to this and have sort of compiled a list of questions, but you are certainly welcome to add more questions as we go through. So you've already explained the phase 1 dose escalation and the biopsy at 12 weeks and 22 weeks, and seeing nothing made this decision. So how many were recruited into the phase 2 or 3 study prior to this decision?
We're not really sharing the number of patients that were exact numbers at this point. I'm unable to share that right now. What I can say is that recruitment was underway in several countries throughout the world. We'll provide a full accounting of that when we present the data.
Okay. And Alex, I'm going to turn it over to you.
Yeah, hi. Thank you. So do you feel that you were able to use the right doses? We did actually hear reports of patients experiencing side effects from the drug. Did that affect your ability to be able to dose high enough? Were you happy with the doses used?
So we actually selected the dose in 2 ways. First of all, based upon the clear results from the preclinical studies with Suvodirsen and a surrogate molecule that looks similar to Suvodirsen, as well as the safety from the phase 1 study. We believe we have the right doses because based on all of that, the doses of 3.5 or 5 should have led to skipping and a translation into dystrophin. Why that didn't work, we don't know, and hopefully we'll figure out how to, why that did not translate. Although we did have, again, fevers in our single ascending dose study, we talked about infusion-associated reactions at the various meetings. Our ability to get to the dose we needed to cause skipping was not hampered by that.
Okay. And was that the only side effect you saw, the fevers, or were there other side effects?
So the side effects we reported in association with our single ascending dose study, and there were no new safety concerns or signals that occurred in this study as compared with the data we've presented recently, most recently in June at the PPMD meeting. All the details will be consolidated and put in a presentation and medical meeting. I think the key thing here, though, is that this was really purely an efficacy decision, and we didn't see any safety signals that would prevent us from proceeding.
Okay, and I think you confirmed this in your opening statement, but some families have expressed an interest in getting access to their individual results. You're going to make that happen?
Absolutely. We've been hearing that from before we even started our program, and that's a commitment we made years ago, and we're going to follow through on that.
Great. Well, we're grateful for that. Within the informed consent, was there a provision for the baseline biopsy to be made available and potentially serve as a basis for other studies requiring a baseline biopsy? Will WAVE allow participants' biopsies to be made available to other companies if participants can enroll into another trial?
So in this study, we tried to minimize the amount of tissue we collected, given the young age of the patients, but also we used the deltoid, which is extremely thin muscle. So we took the most we could, and we used much of it for the analysis, and we're continuing to store any remaining baseline biopsies that was collected. And we're going to use all of those, of course, for the analysis I described earlier, trying to understand what happened. We can't guarantee there would be sufficient tissue for other sponsors to use to do all the tests that they need to, because every study has the same types of requirements that we had. Of course, it would depend upon also whether another sponsor is using the deltoid and whether they would need the sample taken around the time of entry, because there are some challenges of using pooled placebo muscle in a study. However, we're happy to discuss this with another sponsor. We're open to this, but it could be that it just isn't possible. But we're certainly open to a discussion.
Okay, but it's looking like, is it sounding like patients will be able to get the analysis of their biopsies?
Yep, we're going to, all of the data, every patient's data will be provided. It will come through the physician, though, because confidentiality issues prevent us from interacting directly with patients. So we would be providing the information to the principal investigator of the study at the site where the study was conducted, and we would be asking those physicians to share these data with their patients and their family.
Okay, we've got a question here about the samples like serum and urine. Are you going to make those available? The other samples you've collected as part of the trial?
We have not really thought through all of the different possible samples. What we have, we've been focused on the biopsies. We have urine samples and blood samples, an enormous quantity of them for different purposes, and some of them just aren't essentially storable. So we have to go through all of that. We're open to those conversations. We've made no decisions on what we can do with those samples right now.
Okay, and now I've got some questions about the ports. So we'll waive for the removal of ports. Can patients keep their ports if they wish to? Some families are looking into other exon skipping trials now that would require a port, so they'd like to keep the port in for now.
So, what happens with a port in an individual patient, we are completely deferring to the physician. If there's a medical necessity, even for whether it's for treatment with an approved product or whether it's a treatment in a study, certainly the port is at the physician's discretion on whether it be left in, and also there's really no reason why the port couldn't be used for any of those purposes, whether it be a trial or whether it be approved product. If there is no reason to have the port and the physician decides, no, I don't, in the family we don't need the port, of course WAVE will pay all expenses for the port to be removed.
Okay. And as you can appreciate, Pat and myself and many other patient organisations have had, some very difficult, heartbreaking conversations with families this week. What support are you going to provide those families now the trial is finished? Are you going to look at funding things like psychological support?
So, I can understand how this would be, for all the reasons you say, just a traumatic event. As a company, we really don't have a mechanism to do that directly with patients. I think the best way I would recommend is that patients and their families have this discussion with their physician, and then we work with they work with advocacy groups such as yourself and others represented here today to see what mechanisms might be possible through those groups. That would be my suggestion in that regard.
Okay, and then just a question about the process of notification. You know, families were understandably very upset when they found out the trial was being discontinued via social media. For many families, it was hours after the press release that they were able to finally speak to the doctor. Can you just explain the communication process and why that was?
Sure. This is a tough one, and I know it's always a tough one in clinical trials when this happens. And I apologize that many people had to find out that way because we really tried our best to communicate in a way that we could get the information out to the most people possible in the shortest time frame possible. In terms of timing and how these work, we unfortunately have regulatory obligations that make us have to start the process of communication with a general public communication. However, that went out, and within a minute, we then sent a parallel communication to all the physicians involved in the study and all the advocacy organizations, as I said, within a minute of that public disclosure. This is complicated. One could say, well, could you do that with the patients? So this is pretty complicated in that, due to patient confidentiality, we do not have the contact details of patients in the study, and all communication, there are regulations around this, all communication has to be via physicians to get this information out. What we also had to do at the time, which was an additional complication, is that we had to get on the phones to the sites around the world who had patients scheduled for biopsies and port placements and infusions so that we could stop those and that no patient, no child was placed at immediate risk. We did what we could to get the information out there, but it was obviously not perfect. And all I can say is to please believe me that we did the best we could, and certainly people finding out later than they should have is no lack of respect or acknowledgement of the sacrifices made by the families who participated in the study. It really is a logistical, legal work, and we tried to do the best we could.
Okay.
Thank you.
I think Pat now is going to ask some questions on regulatory.
Sure. And thank you, Jeff and Mike, for these answers. And keep in mind, to everyone that is listening in, the only screen slide that you will see is a single screen slide. So you are not missing a series of slides. And I know people are worried about some technical difficulty, but there aren't any slides associated with this presentation. So I just wanted to talk about the regulatory process. You were part of the FDA CID program, which is complex and innovative design, which has all sorts of different aspects to it. And it was really, and it is one of the programs that FDA is really impressed with and working hard to explore. So I'd love to understand some of the nuances of that, what you learned from it, what you can tell us about it, and if you'll sort of give us a high-level explanation of that program and what you were doing inside of it.
Sure. Well, the Complex and Innovative Design Program was our way of trying to address some of the concerns and the learnings of the past few years about what patients want to see in a trial and what they don't want to see in the trial. I think one of the key things that nobody likes to see in a trial is a placebo group. So we worked with FDA. We actually applied for a program where we came up with a method for reducing the potential number of placebo patients required in the Distance 51 study for a definitive result that involved some of the interim analyses that I mentioned to you in my opening statement, and it importantly involved the consolidation of historical data sets from the other companies and academics who had collected this valuable resource from previous studies, in which we were trying to use those patients who had already completed those studies to be matched to our placebo patients that we were actively recruiting to use the historical data to supplement our existing placebo patients and hopefully reduce the number of placebo patients required, and actually reduce the overall number of patients required in the study to show an effect primarily on the North Star ambulatory assessment. So that was the nature of the program. FDA was heavily involved in the design and analysis of the study. Proposed analysis and design of the study because we didn't get there. And one of the commitments under that program is to share all of our learnings from it in public forum and to share it with the community to help the design of studies in the future. In fact, our lead statistician who designed the program is going to be presenting at statistical meetings this year to share some of this information jointly with FDA to show that, again, to try and advance innovative trial designs, especially in diseases like DMD.
Thank you. We so agree with you that the fewer exposed to placebo, the better. And so we appreciate you taking advantage of the CID program, and we'll look forward to the results or your learnings, key takeaways from that program. I know you've also done some work around the patient voice. Can you tell us a little bit about what that program involved and when or if you'll be sharing some of the learnings from that as well?
Sure. I'll turn that over to Jeff for him to comment on that.
Sure. Sure, yeah. Thanks for the question, Pat. Yeah, we've had a lot of, that's been a big effort on our part to understand and incorporate the perspectives of the families into all of our work. And it's been many-fold. There's been multiple activities. And I first just want to thank all of those who have participated with us, who have contributed to that, who have been open, just really open to sharing their perspectives. And absolutely, it's going to be a top priority of ours over the next weeks and months to consolidate that, to communicate that so that the learnings can go to inform the community going forward. Absolutely.
Yeah, thank you, Jeff. I think it is because many of us on the phone today, and certainly in studies that aren't on the phone today, come from very different countries, cultures, and philosophies. And I think that learning about this collective voice is so critically important for our community so that we can participate and that we are valued in terms of our voice and perspective. So thank you for all your work around that. I know that you have not been able to share how many patients were in the phase 2-3, and so I'm just wondering, do you have sufficient placebo data? Do you expect that you could share it with some of the consortia, such as DRISC or the CTAP consortia?
Oh, we have very limited placebo data at this point. We were literally not far into the study, so the placebo data that we have is quite limited. But, we're members of DRISC and we're part of CTAP, and, we'll work with these groups to see if there's some way we can help.
Good. Thank you. Thank you very much. I think it's so devastating to know that you're not going forward at this moment. We really do hope that you can get back to the drawing board and understand where the target engagement, whether it was, in the, as you talked about at the various levels, if it got out of the blood to the muscle, if it got into the nucleus, et cetera, so that we can really advance this opportunity or at least understand why it didn't work. So with that said, I really do appreciate you taking time today. It's really important to our community, and we so value our relationship with you. The World Duchenne Organization and PPMD appreciate this interaction and appreciate your coming forward and talking about the patients and talking about the learnings. And certainly we wish you well. And we're hopeful that we will see you again in the near future once you solve some of the issues that you're going to be looking at in the future. So with that, I'm going to say to everyone on the phone, happy holidays. We are still devastated. We will learn from this, and we will continue to progress. So thank you all for joining us. Thanks, Alex. It's wonderful to hear your voice. And Jeff and Mike, you're wonderful, and we so appreciate your work with us. Don't go away. Happy holidays.
Thank you so much, Pat.
Happy holidays to everyone.
You're welcome. Thank you. Bye.
Goodbye.
Bye-bye.